que tengan para la conferencia de la doctora Saif, las van a ir haciendo a medida que vaya la presentación de la doctora Saif, las van haciendo en chat, así cuando termina tenemos una serie de preguntas para hacerles. Y vamos a dar comienzo entonces a esta sesión especial con la conferencia de la doctora Linda Saif. La doctora Saif ha sido designada como Distinguished University Professor por la Universidad del Estado de Ohio en USA, donde realizó toda su carrera profesional, desde el bachelor en el Colegio de Worcester a su máster y PhD en virología e inmunología. Su expertise es en virología e inmunología, trabajando con rotavirus, coronavirus, calicivirus, y ha realizado una importante contribución en el área de la comprensión del eje lactogénico mamario, que es una de las bases de la inmunidad en todos los mamíferos. Es una de las primeras investigadoras que trabajó en el mundo con el Sars Coronavirus, desde el año 2003. Ha sido convocada desde entonces por organismos nacionales de USA y otros países del mundo, y es una referente internacional en este tema de SARS Coronavirus de la Organización Mundial de la Salud y la OIE desde el año 2003 a la actualidad. Una de las características de la doctora Saif ha sido la formación de gran cantidad de recursos humanos, doctorandos en su laboratorio en USA, de todo el mundo, de Asia, de África, de Europa y de América. En nuestro caso tenemos una particular relación con la doctora Saif a través de los trabajos conjuntos que iniciáramos en el año 86 con su laboratorio y el Instituto de Virología de INTA en rotavirus bovino y en el desarrollo de vacunas eficaces para la prevención de estas enfermedades e infecciones de las diarreas neonatales de los terneros. Ha sido distinguida, tiene numerosos trabajos, como digo, es una referente internacional. Tendría horas para hablar, pero rescato las principales distinciones de la doctora Saif. Fue distinguida por la Universidad de Ghent en Bélgica, es ganadora de muchísimos premios nacionales e internacionales, entre ellos el Wolf Prize on the Agriculture Field. Es la primera mujer que gana este premio en los Estados Unidos y fue designada miembro pleno de la Academia Nacional de Ciencias de Estados Unidos en el año 2017. Nosotros tenemos el privilegio de que haya sido designada académica correspondiente de nuestra academia en el año 2018. Dr. Saif, thanks for your contribution with our academy on this talk, and everything is in your hands.
Okay.
So we listen to your presentation on the SARS coronavirus and emerging coronaviruses.
Okay. So first, I'd like to thank Alex for the kind introduction and the Academy for inviting me to speak today. So it's like going back in time for me to see all my former friends and colleagues in Argentina who I haven't seen for some time now. But today I'm going to talk about COVID-19 and emerging coronavirus and focus on interspecies transmission and then One Health perspectives. And as Alex stated, I'm in the College of Vet Medicine as well as the College of Ag at The Ohio State University. So for those who are not familiar with coronaviruses, there are 4 genera, the alpha, beta, gamma, and delta. And they're positive sense envelope single-stranded RNA viruses, the largest RNA genome, and the spike shown here in the electron micrograph on the schematic encodes the virulence and receptor binding epitopes, and it's the vaccine target for neutralizing antibodies. So all the current vaccines are either inactivated or spike vaccines for SARS coronavirus 2. It infects mammals and birds. It causes mainly respiratory and enteric disease, but it can cause some other syndromes, particularly in different species of animals. All the coronavirus have a similar genome organization. They encode genes for 4 major structural proteins: the nucleic capsid, the envelope, the membrane, and the spike. And in addition, they also encode these in purple here, open reading frames, variable numbers and locations in the genomes. Many of these, the function is unknown, but a number of them code for, these are called accessory proteins that can block the host innate immune response. This slide summarizes the ecology and chronology of coronavirus emergence, and 2 notable features. The ancestral host are bats or birds. So birds for the gamma and delta coronavirus. And then for the alpha and beta coronavirus, it's mainly bat reservoirs. So if we focus mainly then on the zoonotic strains that are transmitted from animals to humans, we will see we have the spillover of the common cold 229E alpha coronavirus from bats through alpacas to humans, estimated in the 17 to 1800s. And then we also recently detected, Dr. Vaslova, myself, and Dr. Gray from Duke, an alpha coronavirus that we called human canine feline canine coronavirus human pneumonia 2018 in children with pneumonia from Thailand. And then we also have the common cold OC43 beta coronavirus. It's thought to emerge from cattle to humans in around 1898. And it's interesting because this was the time Russian flu was circulating. The etiology was never identified, so it's possible this was actually the first coronavirus pandemic. So we have these endemic common cold strains, the alpha coronavirus 229E NL63, that was centuries before these other viruses. And then recently this canine coronavirus human pneumonia strain. For beta coronavirus, for the common cold, OC43 HKU1. So then the next thing that has emerged are these pandemic, epidemic strains. We had the original SARS coronavirus from bats to small camelids, civet cats to humans. This emerged in China in 2003 and then largely disappeared in 2004. But then we also had the MERS coronavirus that emerged from bats through camels and spilled over to humans. And this was almost a decade later. It's still circulating in the Middle East mainly. And then in 2019, we had the emergence of the SARS coronavirus 2. Ancestral host is likely bats. And we don't know if there was an intermediate spillover host to humans. We also have spillovers from bats into pigs, and we have the porcine delta coronavirus that spilled over from birds to mammals, including pigs, and then also humans. And this is shown here. So this is the only cluster that has a large number of both mammalian and avian coronavirus. So the spillover and emergence of new coronavirus has occurred repeatedly in both humans and animals. So I'd like to address a number of questions today. First, why are coronavirus so diverse and how do they emerge and evolve to infect new hosts? Secondly, which coronavirus cross the species barriers? And then what are the potential animal reservoirs or hosts for COVID-19? How are they transmitted from animals to humans and vice versa? And then last, a role of One Health. So the first question, why are these coronavirus so diverse and how did they evolve to infect new hosts? As I mentioned, they have a large RNA genome, the largest RNA virus with high plasticity to accommodate many changes. There are recurrent deletions in the spike, the N-terminal domain that can alter antibody epitopes, but even though coronavirus has a proofreading mechanism, it cannot correct these deletions. They occur as quasi-species or swarm of viruses in a population. And this may explain the origin of interspecies variants. with altered tissue or cell tropism. So I'll give the example here for the swine coronavirus, the enteric transmissible gastroenteritis that mutated into a respiratory strain, the porcine respiratory coronavirus. There are other examples with the feline enteric coronavirus that mutates within the cat to a systemic infection of macrophages that's fatal, feline infectious peritonitis in cats. The viruses, like influenza, also have a high recombination frequency, as high as 25% in mixed infections. So mutations, deletions, and recombination can lead to the emergence of new strains. So I'm going to give some examples of some of these factors in terms of coronavirus. In terms of interspecies zoonotic transmission, example for the alpha coronavirus, we know there's a reservoir community of genetic host range mutants that circulate in cats, dogs, pigs, and then this recent evidence in humans. These viruses all share aminopeptidase N receptors, so they can co-infect across species. Recombination and mutation and interspecies transmission with new strains are common. Here are some examples. All the blue genes denoted here in the genome sequence are from cats. The gold ones are from dogs, and the pink ones are from pigs. So we can see double recombination events that led to a new feline strain with the canine coronavirus spike and a new canine coronavirus strain with the spike from the feline coronavirus. We also know that the coronavirus 2A is the ancestor of TGE, where there were mutations in the spike in open reading frame 3B and deletion of open reading frame 7B. And then more recently, there was a recombination between the swine strain and the canine coronavirus 2A that resulted in mutations in the spike with a recombination here with this N-terminal domain of the spike from the pig strain. And then most recently, both our group and with Dr. Gray at Duke, who surveyed these samples from children that were hospitalized with pneumonia in Thailand, we found this canine-like strain in humans, and it has a mosaic of the spike from swine, cats, and dogs. And the backbone is most like the canine coronavirus strain, but there are mutations in the N and a large mutation in open reading 7. Interesting, another group found a very similar strain in children in Haiti. So this shows there can be host range mutants where the virus can circulate among these species and even spill over into humans. So this allows the virus to persist in these species and evolve in these species. So I also give you the next example about an interspecies variant within species with an altered tissue tropism. And a major example of this is the porcine respiratory coronavirus. And the origin of this, initially, the TGE, transmissible gastroenteritis virus, infects the intestine. Essentially 100% of the villus enterocytes. It causes fatal diarrhea in baby pigs. But this virus that evolved, the PRCV, now infects the upper respiratory tract and also the lung, here infecting epithelial cells lining of bronchi, and atypical pneumonia in the lung, very similar to the SARS Coronavirus 2 lesions. And it's thought that this occurred because of a gene deletion in the spike in this N-terminal domain region, but the receptor region in the S1, the APN binding region, was retained by both TG and PRCV. So the TG and PRCV share the same receptor, but the tissue tropism were thought to differ due to the loss of this sialic acid binding region that's needed to bind to the gut mucins by TG, and this is lost by the spike of PRCV, so this virus no longer replicates in the gut. So the persistence of coronavirus within a host can also lead to variants with altered tropism and disease expression. So we asked the question with a postdoc from China, Dr. Tsunemitsu, in 1995, do beta coronavirus cross the species barrier? So to look at this, we use germ-free calves that don't have any coronavirus or any other microbes and also are antibody-free. And we also got a human strain of an enteric coronavirus from a child that was very similar to bovine coronavirus. And we isolated coronavirus from sandbar deer, white-tailed deer, and water bug. So we inoculated each of these into our germ-free calf. All the calves got diarrhea with all these different viruses. They all shed the virus in feces, and they all seroconverted with neutralizing antibodies to bovine coronavirus. So this is one of the first examples of the spillover from wildlife, wild ruminants, through cattle, and also likely two-way spillover, first into humans and then humans back to cattle. So this is very relevant now because in recent studies, the SARS coronavirus 2 has been detected in white-tailed deer in the Midwest and also the eastern part of the US. So 2 studies showed that white-tailed deer are susceptible to infection with both the original SARS coronavirus 2 strain and the alpha coronavirus variant of concern. This is the alpha, beta, gamma, delta, and so forth variant of concern labels. So antibodies to SARS coronavirus 2 has been detected in an average of 40% of US white-tailed deer, and in some states it was high as 60%. And then recently we collected deer samples from January to March 2021. We tested these in RT-PCR, the nasal swabs, and we found over a third, 33% of Ohio deer actually were RT-PCR positive for SARS coronavirus 2. So we're now investigating the genomic sequence of these, but likely these were all spillovers from human cases. So another question then is what factors influence the interspecies transmission of these enteric and respiratory coronavirus? So one is environmental exposure, the host reservoir interactions. So we need close contact with infected animals, infectious secretions or fomites. This can occur at the wildlife, domestic animal, human interface, especially when there's interaction with bat reservoirs, bat caves, or bat droppings. Also, an ideal situation for this is the live animal markets in Southeast Asia, where there are multiple species, unsanitary conditions. And then porcine epidemic diarrhea coronavirus in swine that killed about 8 million baby pigs in the US was thought to have come from China because the virus sequence was most like the Chinese strain. And it's postulated that it may have come from fecally contaminated feed sources from China. We also know there can be environmental contamination, such as wastewater, sewage. And so this is often monitored for a warning about what coronavirus, SARS coronavirus 2 variants are circulating within a community. Another factor is stability or dose of the agent in infectious material, the transmission route. It's possible there's a greater efficiency of combined aerosol and fecal oral transmission, since these are really these beta coronavirus are pneumoenteric virus. They infect both the respiratory tract and gut. And these are the bovine coronavirus, SARS, MERS, and SARS coronavirus 2. The viral factors include the attachment entry receptors, co-receptors. We know that like SARS coronavirus, the SARS coronavirus binds to the ACE2 receptor, actually with higher affinity than for SARS. This is shown schematically here on the cell and in the schematic where the spike binds to the ACE2. And also the SARS coronavirus 2 has a new furon cleavage site that splits S1 and S2. And this is through the TMPRSS protease on the surface to allow the virus to enter the host cell. So some of the first observations about SARS coronavirus zoonosis in 2002, 2003 were that workers in the live animal markets in China had antibodies to SARS coronavirus 2, but no disease. So the animal host, when the markets were surveyed, the SARS coronavirus 2 RNA was found in mass palm civets, a very high identity to the SARS in humans, but also raccoon dogs, but not in the wild or farmed civets. So this was happening in these caged animals in the marketplace. After the SARS outbreak, the sequence data confirmed that certain bats, these Rhinolopus horseshoe bats, harbor SARS-related coronavirus. And then after that, it was found that some bat strains use the human SARS coronavirus 2 receptor ACE2, and that suggests there could be a role for bats in direct transmission of these SARS-related viruses to humans. So in one publication, there was an indication that SARS-like coronavirus in bats are a time bomb for the next global pandemic. But these warnings, unfortunately, were largely unheeded. So the COVID-19 zoonosis, again, the observation is some of the early cases had contact with live animal markets in Wuhan, China. And in fact, there were two lineages, lineage A and B, and it's thought that these may have spilled over different times into humans. So the bats are the likely host ancestor for SARS coronavirus 2, again, mainly these Rhinolopus bat species. So it's now shown that multiple Asian bat coronavirus are related to human SARS coronavirus 2. So these viruses from China, designated here, this one in 2013, share high overall genome identity, but these have low identity in the receptor binding domain. And now we have viruses from Cambodia, from Thailand, that also were found from Rhinolopus bats that share a high nucleotide identity, and some of these have insertion deletions at the S1, S2 furin cleavage site, even though they do not have that particular site. But more recently, there were 3 strains found in Laos that share the highest identity to the spike receptor binding domain of SARS coronavirus 2. They only differ in 1 to 2 amino acids, and they have a very high efficiency of binding to human ACE2. So again, this suggests the role of bats in the possible direct transmission originally of SARS coronavirus and possibly now SARS coronavirus 2. Some people also suggest there's still an intermediate host though, such as pangolins that may have transmitted the virus to humans from the bats. But for the pangolins, although there's a high identity in the receptor binding domain, overall, the genome identity is less than for the bat strains. Other possible intermediate hosts could be carnivores, like for SARS-2, or muscelids, which are highly susceptible to SARS coronavirus 2. So now we have encountered another situation where mink and pets could possibly be secondary hosts for COVID-19. So this originated from studies of farm mink in Europe and also then in the USA and Canada. There was a rapid transmission of SARS coronavirus 2 among mink, so millions had to be euthanized. Interestingly, the disease in mink is very similar to the disease in humans. The mink showed respiratory signs, shed virus, nasal secretions, some had interstitial pneumonia, and some died. More concerning, there was 1 case in Utah and 2 cases in Spain, where there was likely spread from mink farms into wild mink. And then there's been documented evidence of repeat infections in mink, just like can occur in humans. And after these infections, antibody titers were boosted in the serum. So the thought is originally humans, the caretakers, transmitted the virus to the farm mink, but then there have been evidence from Denmark and the Netherlands that the mink could transmit the virus back to humans. So in Denmark, 214 human SARS coronavirus 2-like infections were detected with mink-related variants, and one cluster, cluster 5 variants, had 4 genetic changes in the spike, with 1 prominent in the receptor binding domain. So this raised concerns for effects on transmissibility, diagnostics, and vaccines for humans. As I mentioned, there's also potential spillover into wild mink, and in 1 mink farm, cats were also, stray cats were shown to shed SARS coronavirus 2, presumably from the mink. So the question is, will vaccines be needed in the secondary host to prevent new variants and spillovers? And one scenario that I showed here the mink is a certain case, particular case, but now we also have it in wildlife in deer in the US. So this is of concern. So others have suggested pets may be secondary host. So pre-viral emergence, we had no evidence of transmission from pets back to humans, but dogs and cats were infected in households of COVID-19 cases, at least 135 pets in the US. And then cats were shown to be experimentally susceptible in 4 different studies with viral transmission to contacts, respiratory lesions, and seroconversion. However, dogs were only marginally susceptible, but they did seroconvert. So now we're in the post-variant emergent stage of SARS Coronavirus 2 circulating in humans, where we have various deletions or mutations in the spike. And here, this is shown for the spike receptor binding domain and the receptor, ACE2 receptor. So we have alpha variants from the UK, beta variants detected originally in Brazil, and gamma in South Africa, and gamma in Brazil. So we don't know if these variants will transmit better in humans because all of the early studies have been done with the original strains. But it was shown in the UK that the alpha, the B117 variant, was detected in dogs and cats from infected homes, but also in Italy and the US. And these pets in the UK had an increase of myocarditis, but we don't know if that's cause and effect or simply circumstantial. Now, it's also of interest that neither the original strains, the B1, nor the alpha variant infected the laboratory mice, but these variants of concern, the gamma P1 from Brazil and the beta B1351 from South Africa, could replicate directly in the lungs of lab mice, suggesting the variants can show a different pathogenesis and susceptibility pattern than the original strains in animals. So now we have COVID-19 and a number of experimental or naturally infected animals. So all of these in green are highly susceptible to SARS coronavirus 2 infection, either directly or direct spillover or by experimental infection. And this includes wildlife, various wildlife that are susceptible. The ones in white are moderately susceptible, including cattle and dogs for experimental studies. And the ones in pink have been shown to be not susceptible, but the lab mice were susceptible to the variants. So because all of these spill over from humans to date, it's very important to take a One Health approach to controlling these, to monitor not only human health, but also animal health and environmental health. It's of interest that for some of the zoo animals, the mustelids, the primates, and the felids, it's been shown that some of these now are picking up the virus, presumably from the human caretakers. So Zoetis developed a spike adjuvanted vaccine that's now being tested in zoo animals and will be used in zoo animals. So to summarize, in terms of coronavirus interspecies transmission, we have bird reservoirs for Delta and Gamma coronaviruses. And here we can see these large feed bunks in the Midwest. This is open corn feed bunks here, where we see these are Canadian geese. And on the right, these are songbirds, mainly starlings. And it's potentially possible that since we had this porcine Delta coronavirus arise in the US in 2014 associated with diarrhea and vomiting and some deaths in baby pigs, whether this could have been from the feed, because these are the animals' feces is in the feed that's fed to the animals. We know that for the alpha, beta, clade B and C coronavirus, they're bat reservoirs, these rhinolopus bats mainly. And then for ruminants and rodents are likely reservoirs for the beta A bovine coronavirus group. So why bat and bird reservoirs? There are multiple species, they're high density, and they're migratory. So they can carry the virus subclinically because they do not show disease long distance. So this suggests coronavirus adaptation to new hosts with interspecies transmission can expand in the species with interspecies variants, and multi-species transmission can establish a new host reservoir community. And this is a concern now for COVID-19. So I showed you, just to summarize, the alpha-1 species host reservoir community where the virus circulates among cats, dogs, and pigs with new recombinants that can spill over into these species and occasionally apparently spill over into humans. Has only recently been detected. For the Imbeco beta coronavirus 1 species, cattle, it's been shown the virus can spill over between cattle and cervids, the deer, white-tailed deer, also experimental transmission from a bovine coronavirus strain to turkey poults, back and forth to humans. And then by genetic analysis, we have very similar viruses to the bovine coronavirus that are thought to be from this lineage in pigs, horses, and dogs, this associated with canine kennel cough. For the Sarbeco beta coronavirus, COVID-19 possible reservoir communities now, obviously we have humans that probably likely from bats, possibly through an intermediate host, but now we've shown there's spillover, two-way spillover mink, at least one-way spillover into deer. And the concern is once it's in deer, whether it could spill over into livestock who graze on these pastures that are populated by deer, cattle, sheep, goats, or into other wildlife species. And also we have the cat spillover into cats and dogs. So WHO has predicted the next pandemic will be disease X, and not if, but when there may be future coronavirus transmissions to humans. So what are some of the concerns or the most likely strains for spillover? We have these SARS-related beta coronavirus in bats. They've been associated with 3 prior pandemic epidemic events. We have the SARS, the MERS, the SARS Coronavirus 2, as noted here, and some SARS-related and SARS Coronavirus 2 related bat strains combined directly to human ACE2. Another scenario that's of concern, there's a swine alpha coronavirus, severe acute diarrhea syndrome epidemic in southern China, killed about 25,000 baby pigs. And that virus is most similar to a virus circulating in bats near the pig farms. It's also been shown this virus can infect primary human lung and intestinal cells. The third worrisome coronavirus is the porcine Delta coronavirus discovered in pigs in Hong Kong in 2012. It spread to the US as an epidemic in 2014. It has a broad host range. We know this porcine Delta coronavirus can infect turkey poults. It caused a mild infection in germ-free calves. But now there's reports from Haiti of 3 Haitian children with mild febrile illness with these spillover porcine Delta coronavirus strains. It also infects human cell lines. It shares an aminopeptidase N receptor that's shared among these species. And it's likely had a bird, songbird host reservoir. So the concern here is an influenza scenario where you have spillover from avian species into an intermediate swine host, and then transmission from the intermediate host to humans or from the bird host to humans. So to summarize, coronavirus continue to evolve and emerge globally as a cause of fatal respiratory and enteric infections in young animals and humans. They're genetically diverse with frequent mutation and recombination. Some beta coronavirus have broad host ranges and are zoonotic. The epidemiology and zoonotic transmission of SARS Coronavirus 2 infections are unclear, as is the potential for reverse zoonosis or spillback from the animal strains back into humans. and that would establish a new COVID-19 host reservoir communitized humans. So what can be the One Health focus to try to mitigate coronavirus pandemics? Certainly surveys in animals, prioritize target animals, certainly bat species, but the small carnivores, muscelids, and now cervids might need to be surveyed. Environmental surveillance through wastewater sewage, environments with contact with infected animals, infectious secretions or fomites, as I mentioned before, the bat interface, but also live animal markets, possibly fecally contaminated feed sources for animals, human surveillance for infections by animal coronavirus, respiratory enteric infections, not only symptomatic hospitalized patients, but probably outpatient clinic, the immunocompromised. We may need to develop animal vaccines to prevent new animal host reservoirs. And then finally, developing funding mechanisms to support integrated collaborative research on zoonotic diseases, including both the human and animal reservoirs. So we need this One Health focus with the combined efforts of veterinary for animal health, medical for human health, and environmental scientists with cross-disciplinary interactions and global interagency cooperation. So I'd like to acknowledge my lab, my former postdoc, Dr. Vaslova, who's now an assistant professor, has her own lab, former colleagues who contributed to this work. Dr. Chiang Wang Lab was working on PD and porcine delta coronavirus. She's a former grad student, now associate professor, and then a new assistant professor, Dr. Scott Kenny, working on porcine delta coronavirus. And our funding agencies. And then just to reminisce, all my former excellent postdocs, visiting scholars, and students who have come from Argentina. And with that, I'll be glad to try to answer any questions.
So thank you very much, Linda. Excellent presentation.
Thank you.
A lot of work. And well, let's go for the questions, if there are any. There is any. Let me see. Chat. I don't see any question in the chat, so while we wait for the questions, I was thinking in many of the aspects of your presentation. Two main things, Linda. One is related with the eco-evolution of this type of viruses. And it's very general. You have mentioned that for the alpha server and MB group of viruses or variants in which you consider the environmental distribution, only in the server the bats appear as a reservoir at present time. My question is, there is a common evolution of the SARS coronavirus from a common ancestor in bats, or none of them have relationship? They have make the variation to adapt to these different environments after from the SARS coronavirus itself. Please, if you, I don't know who, but this close your microphone, please. I don't know who is, but close your microphone, please.
So
okay.
Pues no veo.
Summarizing the question. Summarizing the question. There is a common evolutionary tree from a bat coronavirus reservoir, or they have different, they have differentiated after.
Okay, so there's apparently a common reservoir host in bats, particularly the Rhinolopus species, but there are large, I think the bats are the second largest group of mammalian species with different species. So there are a large number of bat species, but the distinctiveness seems to be for these viruses that have evolved in the Rhinolopus species. And the fact that they have the most similarity to SARS and SARS Coronavirus 2, but more importantly, some of those viruses also can use the human ACE2 receptor to infect cells. So that's the concern, that some viruses that are present in bats could spill over directly to human, the SARS-related bat strains and the SARS Coronavirus 2 related bat strains.
Okay.
But other bat species have their own repertoire of coronavirus. Coronavirus are very common in other bat species too. I mean, it's thought that some of the animal strains, like porcine epidemic diarrhea virus, originated in bats. Some of the common cold strains originated in bats, potentially other species of bats. But the other commonality of the bats, because there are different species that co-inhabit the caves and the habitat, then these viruses can recombine in these bat species. So you can have new strains and mosaic viruses in the bats that then may have more ability to spill over into other mammalian species. So because bats are mammalian species, it's quite likely it may be easier for the viruses to spill over from bats than say avian species. So there's definitely a major repertoire of coronavirus, different coronavirus strains in different bats.
Okay, thanks. There is a question here from Bratanich. How important is MERS as a potential cause of a pandemic event?
So it bears watching, because the problem is we don't have good predictability for these coronaviruses about exactly what it takes for them to become a widespread to cause a pandemic. So what's happening in the Middle East right now is there seems to be multiple introductions from camels into humans, and this keeps continuing because the camels are now a reservoir for the MERS coronavirus. But the virus has not yet acquired a great ability to spread from human to human. But the more the virus circulates, the more opportunity it has to change and to acquire that. So this virus should be closely watched as well, because we don't know when it might acquire more human to human spread ability, because we don't understand that yet exactly all the factors that allow this virus to spread so efficiently in humans, like SARS Coronavirus 2. I think SARS Coronavirus 2 can provide a blueprint for that, about things that we need to watch and monitor, but we don't know that yet.
Okay. Another question from Dr. Caposo. What is your opinion of using serology to screen for susceptible species in the field? Can it be interfered by antibodies that cross-react with other coronaviruses? Is there any information about that?
Yeah, so this is a very good question. So in 2003, we set up a big collaboration through WHO to supply animal coronavirus antiserin strains to the whole coronavirus research community. And when we were investigating this with the CDC, we found there was an essentially unidentified cross-reactivity with anaserum to TGE coronavirus and SARS coronavirus 1, which you would not expect between the beta and delta coronavirus. So it could have been essentially something related to a protein and how the protein was folded into this kind of epitope based on the protein folding. But anyway, so we had to design nucleocapsid specific antibodies that reacted only with SARS, that we could delete all the regions that cross-reacted. So that can be done, but you have to monitor carefully for cross-reactivity. So the gold standard so far for monitoring animal sera has been virus neutralization, and most of this, because it's more cumbersome, it has to be done in BSL-3, is done with S pseudotype viruses that can be used in BSL-2 conditions. Another way, the test that was used for the deer samples was an ACE2 blocking assay. And so that was shown to be specific in terms of not cross-reacting with other coronavirus. So the point is that if you don't survey all these other coronavirus for cross-reactivity, you wouldn't know that they're going to cross-react with some other unknown beta coronavirus or alpha coronavirus, delta coronavirus, and so forth. So this is something that really needs to be done, and needs to be done carefully, and needs to be standardized to be sure that serology is specific, but it can be done. Okay.
Okay. Another question is, Linda, is there enough evidence of fomites or fecal oral transmission between animals?
So at this stage, we don't know. It appears we don't know how the virus got into the deer population in the US. It's likely spillover somehow from humans, whether it could have to do with wastewater, because we know we can find SARS coronavirus 2 in wastewater. One would think the dose would be very low, but we don't know how it spilled over. But most of the samples that have surveyed have been nasal secretions, but we also know the virus replicates in the upper respiratory tract and saliva. We also know with SARS coronavirus, about 50% of people can have gut infections, so it can be shed in the feces, and that's why one finds it in wastewater. But as far as between animals, when these studies were done in terms of direct transmission, they were just done by putting sentinel animals in with the infected animals. So the actual mechanism of transmission wasn't identified, presumably nasal secretions or fecal material. But that hasn't been exactly identified in terms of the experimental studies with the sentinel animals and transmission.
Okay, Linda, just to let you know, there are some messages from the group of attendants from Ohio State University, and also from Brazil, from the Brazilian Academy of Veterinary, that congratulate and thank you. And let's go back to some questions. Dr. Sonsini from Brazil, are there any relationship between COVID-19 and the infectious bronchitis virus of the avian species?
So the answer is no, none has been demonstrated. So this virus, the infectious bronchitis virus, is a gamma coronavirus, a different genera. And there's usually not cross-reactivity with viruses within different genera, but there can be exceptions, as I indicated, for the alpha coronavirus TGE and the beta coronavirus SARS coronavirus 2. But if you have serum against infectious bronchitis virus, there's no evidence that it interacts with SARS Coronavirus 2 or can neutralize SARS Coronavirus 2.
Okay, Linda, let's change the scope now. Dr. Palma would like to know from you your opinion. What do you think about the repeated vaccinations with the vector vaccines carrying different antigens? Could it present a problem in the future, or there is no problem?
So right now, the vaccines are mainly carried, other than inactivated, all the vaccines carry the spike antigen. And they carry the spike antigen of the original virus. So far, even though the neutralizing titers are lower, it's been shown there still can be protection against the variants, especially against death and hospitalization. So it's important that even though those titers may be lower, they still can prevent hospitalization and deaths in vaccinated people. So the one concern for the future would be the same concern as with influenza, and that would be this question that's called original antigenic sin. If initially you're primed to one virus, if your immune system is then programmed to respond better to that virus than to a new variant or the next virus. So for that reason, some people are thinking that maybe the vaccines eventually need to be updated to have the spike of some of the variants included. But there were studies done in animals so far where in non-human primates, they inoculated them with the messenger RNA original virus versus a beta variant spike, and then they challenged with the beta variant. And in both groups of non-human primates, they were protected against the beta variant. So until the virus better escapes vaccines, current vaccines or neutralizing antibodies, that's when we probably really need to look at updating these types of vaccines. So I hope that answers the question. For my way,
I think
my way of thinking.
You did it.
Yeah, I think, you know, these are first generation vaccines, and new generation vaccines will be coming. And from my standpoint as a mucosal immunologist, I think we would like to see some mucosal vaccines, intranasal vaccines to protect both the upper and lower respiratory tract. These vaccines all do a tremendous job of protecting the lung, the lower respiratory tract, so we don't have hospitalization and deaths, but they don't stop the virus necessarily from spreading and transmitting. So we hope that if there were an intranasal vaccine, we could actually block virus transmission as well as disease.
Another question that was given to me is with regard to the new variants of SARS Coronavirus 2 that has been detected lately. Are there any evidence from seroneutralization of not complete cross reaction, or do they cross react properly with the available vaccines?
So far, as I mentioned, the titers are much lower, especially the beta variant have the lowest titers, neutralizing titers from the vaccines. But so far, all of them have some level of neutralization. But what's happening is the elderly and immunocompromised people have the lowest, usually the lowest response, even to the initial vaccines that wane over time. And so that's why now for those people, they're suggesting that a booster vaccine may be necessary to maintain the antibody, neutralizing antibody levels. It's thought that especially CD4 T cells may also play a role, or CD8, but that's not clear at the present time exactly what role they could play. They are mainly important in stopping the infection once it's established. The neutralizing antibodies are what block the infection from being established.
Okay, two last questions. One is related with the, you mentioned something, but to put it clear, do you think, or do you have any evidence of development of oral or respiratory vaccines now in course?
Oh yeah, there's some 230 vaccines been developed and a lot of them in at least phase 1 trials now. And quite a number of those, including some of the adenovirus vectored vaccines and the subunit vaccines are now in clinical trials. So we'll see how well those work. All our work in animal, rota, and corona vaccines suggests that you really need a live replicating, live attenuated virus vaccine, the prime, followed by intramuscular booster vaccines are fine for boosting, but priming is a difficult scenario here to protect against both infection and disease.
Okay. Thanks, Linda. If there is something else that you want to say?
No, I just thank all my friends and colleagues in Argentina. It's nice to see a lot of you, even if it's virtually. And it's been an outstanding collaboration. And I thank my fellow Argentine Academy members for attending. And hopefully we can all stay connected even through these difficult times.
Okay. Okay, thank you again, Linda. Best regards for you and family.
Thank you.
And we close.
Thank you. Bye-bye, everyone.
Thank you very much for all attendance.
Thanks for attending. Bye, Anna.
Bye-bye.
Yeah, bye, Alex. Take care.
Bye-bye.
Bye-bye.
So, Karina, close the... cierre la grabación, Karina, y muchas gracias a todos los que atendieron esta magnífica conferencia. Si algún comentario quedó sobre los chats que estaban, lo siento, pero nos estábamos pasando de tiempo. Muchas gracias a todos y hasta la próxima.